starting with proteins. What do you address a protein as? Protein is otherwise called as what? Polypeptide chain, right? So what are the building blocks of the proteins? Amino acids are the building blocks of the proteins. How many different types of amino acids do you see? 20 different types of amino acids. So you see 20 different types of amino acids. So different types of amino acids will make one polypeptide chain, right? Which are not similar, not identical. That is the reason always a polypeptide chain is a heteropolymer. It will never be a homopolymer. Okay, polypeptide chain is always what, ma? Heteropolymer. It is never a homopolymer. Got it right? Protein or polypeptide chain made up of amino acids. Amino acids are the building blocks. We have different set of amino acids, so it is always a heteropolymer, never a homopolymer. This is the basic image that you have to fix it in your mind, ma. First look at a single amino acid. How an amino acid is formed? At the center there is an alpha carbon, to which you have a carboxyl group, amino group and hydrogen and then functional group. With what is the amino acid varying? With the functional group, with respect to functional group. Okay. So these three are common: carboxyl group, amino group and hydrogen is common for all the amino acids. The only variable part here is functional group. Okay. Likewise, based on that, amino acids are classified into two different categories: hydrophobic non-polar amino acids and hydrophilic polar amino acids. This table should be with everybody. Okay. You will get questions either in botany or in chemistry. Okay. So amino acids are of two types: hydrophobic non-polar amino acids and hydrophilic polar amino acids. And in them you have positively and negatively charged amino acids. If you see hydrophilic polar amino acids, negatively charged will be what? Aspartic acid and glutamic acid. Positively charged includes lysine, arginine and histidine. Lysine and arginine are familiar terms to you because they are the compositions of histone proteins, right? So these two are clear, no ma? Hydrophilic polar amino acids have negatively charged. Examples include what? Aspartic acid and glutamic acid. Positively charged includes lysine, arginine and histidine, right? And then positively charged, along with this again few more positively charged polar amino acids include serine, threonine, aspargine, glutamine and cysteine. This is about polar amino acids, beta. Now non-polar amino acids include two types based on whether it is aliphatic or aromatic. Aromatic amino acids are only three, easy to remember, includes phenylalanine, tryptophan and tyrosine, right? Aliphatic amino acids which are non-polar includes glycine, alanine, valine, leucine, isoleucine, proline and methionine, right? This chart should be with all. So just now, even in a single glance, you can remember polar negatively charged aspartic acid and glutamic acid. Non-polar aromatic amino acids includes phenylalanine, tryptophan and tyrosine. Second or third time you are looking at this flowchart, you will be thorough with positively charged polar and aliphatic non-polar amino acids as well, right? Clear? Just go through this chart for a minute. Done, ma? Once it is done, please let me know. we go with the amino acids which are given in our NCERT. Glycine, if you see in the R group, what is there? Only hydrogen group is there. Alanine you can see methane group is there, methyl group, and serine you can see ethyl alcohol. Okay. So these are the functional groups that you have under glycine, alanine and serine. You have to just remember these groups. Okay. And here we go. We have two types of amino acids. The classification is based on polarity, we have seen. Amino acid classification based on polarity we have seen. Now amino acid classification based on the essentiality, that essential amino acids are those which we consume in the food on the daily basis. Non-essential amino acids are built in our body itself. We are not consuming it. Okay. Like that, you remember any one set, the other you can choose obviously. Like this you can remember with a mnemonic, private Tim Hall. Okay, which includes phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine and lysine. Private Tim Hall. You just remember this for essential amino acids. Okay. So just repeat what are the essential amino acids. Phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine, lysine. That's all. Okay. So this is about essential amino acids and these are non-essential amino acids. So you are clear on amino acid classification, right? Based on polarity and based on essentiality. Now we are looking at the charge of amino acids. We have looked on positively charged, negatively charged amino acids, but in a neutral solution, okay, if at all an amino acid has both positive and negative charge over it, a single amino acid in a neutral solution, if it is having both positive and negative charge over it, it is addressed as what? Zwitterion. Okay. If that solution, by reducing the pH, it turns to acidic environment, then it turns to form what? This Zwitterion forms what? Cation. In objective perspective, what you have to remember? If neutral solution, in neutral solution it is as Zwitterion with positive and negative charge. If you are reducing the pH, it will turn to what form? Cation. If you are increasing the pH, it will turn to what? Anion. Right? So you are clear with this, right? Zwitterion is amino acid where it has both positive and negative charge at neutral pH. If that solution pH is further reduced, this Zwitterion will turn out forming what? Cation. If pH is increased, then it will turn to form anion. And you have to remember one more point, that is on net charge. Net charge is positive at low pH. If the pH is reduced, net charge will be what? Positive, that is +1. It is cation and it is positive. If the pH is increased, it turns to form anion and the charge is -1. At Zwitterion, the charge will be what? 0. Okay, clear with Zwitterion? Classification on polarity, on essentiality, third is Zwitterion. This you are clear with. About amino acids, the story is crystal clear in our mind, right? Now, on the structures of protein, okay, we have primary structure, secondary structure, tertiary structure, quaternary structure. You have to remember at least one example of that and what are the bonds involved in that. Okay, so two amino acids are connected by what bond? Peptide bond. The same story is happening here. Water molecule will be removed out and this is how they are connected. C double bond O is connected with NH and this forms out what? Peptide bond. Like how in N-glycosidic bond you are losing one water molecule, right? Hydrolysis is happening. Same the case here. Okay, but then this is happening between carboxyl group of the first amino acid with amino group of the second amino acid. That you have to remember. In the first amino acid, what group is involved? Carboxyl group. In the second amino acid, what group is involved? Amino group, right? And this forms out a peptide bond. Okay, here we go. So the levels are primary, secondary, tertiary and quaternary. Likewise, when from the first amino acid, carboxyl group is involved in bond formation. Which bond will be free? Which group will be free? Amino group will be free, and this is known as N-terminal. Like that, a set of long chain of amino acids are formed. In the last amino acid, which group will be free? Carboxyl group will be free. So this is addressed as what terminal? C-terminal. Okay, first amino acid is having N-terminal, last amino acid is having C-terminal. And how the peptide bond is formed? Connected with carboxyl group of first amino acid with amino group of the second amino acid. Clear, right? Now let us look into each structure. First, primary structure. Look at the image, then we will look into the theory. So at this left end, what do you have? N-terminal, and here you have C-terminal, first amino acid and last amino acid. This forms out a long chain. What is this structure giving the information about? Nothing else, about the position. For example, we have glycine, we have arginine, we have histidine. Let's say these are the positions: lysine, arginine, histidine. Like that, they are in the long thread. Let us suppose that. Which amino acid is first and which amino acid is second, third, like that. You will only get the positional information from the primary structure of proteins. What do you get? Positional information about amino acids. Look at the theory. These are your NCERT liners. The sequence of amino acids, that is the positional information in protein, which is first amino acid, which is second, and so on, is called as what? Primary structure of proteins. A protein is imagined as a line like this. The left end is represented by first amino acid, and the right end is represented by what? Last amino acid. First amino acid is also called as what? N-terminal amino acid. Last amino acid is called as C-terminal amino acid. Clear all? Yeah. So this is about primary structure of proteins. Now, secondary structure you look into. In proteins, only right-handed helices are possible. Listen first. So, primary structure is like a line. You have understood. First is having N-terminal, last is having C-terminal. They are binded by peptide bond. This information is clear to us. Secondary structure can either be in an alpha helix model or it can be as beta sheets. Both are what? Secondary structures of proteins only. Both what? Alpha helix and beta plated sheets. In alpha helix, in primary structure you have seen one amino acid connected to other by peptide bond. But here one carboxyl group of one amino acid is connected to the amino group of 4 down amino acid. Okay. Here also you see carboxyl group is connected to what amino acid? 4 down amino acid. Like that, it is rounded in what fashion? Right-handed fashion. This is how the bond is formed in what? Alpha helix of secondary structured protein. In beta plated sheets, 2 amino acids, they will be connected right away to each other like this. Carboxyl group, amino group, carboxyl group. Like that, 2 chains will be connected. Got the point? This is how alpha helix and beta sheets are formed. Now, alpha helix, look at the theory also. In alpha helix, carbonyl of one amino acid is hydrogen bonded to amino hydrogen of amino acid. This is 4 down the chain. Okay, and this pattern of bonding pulls the polypeptide chain into what structure? Helical structure. This resembles like what? Curled ribbon. Okay, and how? What kind of helical structure you can see? Right-handed helices. Alpha helix is clear to all, no? Now, beta sheets, beta plated sheets, 2 or more segments of polypeptide chain line up next to each other and they will resemble like what? Sheet-like structure. That is why they are known as what? Beta plated sheets. Clear on alpha helix and beta plated sheets? Everybody, secondary structure is clear, no? So in primary structure you can see only peptide bond. Here along with that, what is there? Hydrogen bond is also present. Okay. Next. In addition to this, we have tertiary and quaternary structures. Look at the tertiary structure once. So, in secondary structure, you can see either right-handed helix or beta plated sheets. In tertiary structure, you can see the woolen ball-like structure. The protein is composed to what? Some woolen thread or ball-like structure that resembles to a tertiary structure. So, here if you see, in addition to the long protein chain is also folded upon itself, forming a hollow woolen ball. This hollow woolen ball itself is addressed as what? Tertiary structure. Nothing like alpha helix, beta plated sheets further binds in amongst itself and turns out forming what? Tertiary structure of proteins. And R group includes what type of bonds? In secondary structure, you have hydrogen bonds. In tertiary structure, few more bonds are present, because of which it is coiled into a woolen ball. Okay. So, what are the other bonds included here is hydrogen bonding, ionic bonding, dipole-dipole interactions and hydrophobic interactions. For your understanding, it is very much important to note this. Please take down. Proteins, primary structure includes what bond, ma? Only peptide bond. Secondary structure along with this includes what? Hydrogen bonds. This gets further complexed and turns out forming what? A woolen ball-like structure, that is tertiary structure. Which includes hydrogen bonds, ionic bonds, dipole-dipole interactions, hydrophobic interactions. Done? Now, we look into the quaternary structure of proteins. Quaternary structure of proteins, best example we can take is hemoglobin, which has 2 alpha subunits and 2 beta subunits. Quaternary structure includes all the layers, all the structures of proteins. It has alpha helices, it has beta pleated sheets, it has straight chains, few of the straight chains of proteins. So, it is a combination of what? All the structures. Likewise, this is a structure of hemoglobin, 3D structure of a hemoglobin, wherein you have heme at the center and iron, right. So, some proteins are an assembly of more than one polypeptides or subunits. Hemoglobin is the best example for quaternary structure. It has how many subunits? 4 subunits, which includes 2 alpha and 2 beta sheets. Can you see? This is how they are formed into the hemoglobin structure. Okay. So, it is as simple as it is, no, about polypeptides. Now, this is important directly from your NCERT. From this table, 2 to 3 PYQs I can show you. Proteins are present in n number of forms in our body. Collagen is the abundant protein in the animal world. Okay, it forms out what? What is its function? Intercellular ground substance. Trypsin, there are many enzymes which are proteinaceous in nature. Enzymes are proteinaceous in nature. One example given here, trypsin, which is an enzyme. Hormones are proteins. Insulin is a hormone. Example. Antibodies are what? Again proteins. They fight against infectious agents. Receptors are proteins which senses the reception like smell, taste, hormones, etc. GLUT4, which enables the glucose transport into the cell. Even in cell, the unit of life, we have learned about carrier proteins, channel proteins. So this is the diversity of proteins in our body, right? But these examples and functions you have to remember. Okay, collagen, trypsin, insulin, antibodies, receptors, and glucose transporter GLUT4. Next, look at the functions. This is what is explained here. What is the world's abundant protein? Rubisco. Abundant protein in animal world? Collagen. Okay, and these are the diversity of the functions. Look at the statements once. Some proteins transport nutrients across the cell membrane. Some are hormones and enzymes. Some fight infectious organisms, for example, antibodies. Collagen is the most abundant protein in the animal world. Rubisco, that is ribulose 1,5-bisphosphate, carboxylase oxygenase, is most abundant protein in the whole biosphere. Right? So, with this the proteins will be ended up. So, what we have learned from proteins, if you are taking up, please take. Yeah. Listen now. What we have learned from proteins? First thing, amino acids. Classification of amino acids based on their hydrophobicity, polar and non-polar, and then based on their essentiality, essential and non-essential amino acids. After then we have looked into zwitterion, then structures of proteins, primary structure, secondary structure, tertiary and quaternary structure. At the end, the diversity of proteins that we have in our body, and the NCERT liners you have from proteins, right? So proteins clear to everybody, right? And we are yet to do with two more biomacromolecules, nucleic acids and lipids. Nucleic acids, you see, you will get the questions connecting with mol bio. So listen carefully, right? So nucleic acid is addressed as what? Proteins, like how you can say it as a polypeptide chain. Sugars, like how we are calling it as a polysaccharide. Like that, what you will address a nucleic acid as? Polynucleotide chain. It is addressed as what? Polynucleotide chain. Means it is the chain of what? Nucleotides. Okay. If I have to take a single nucleotide, it is composed of what? Single nucleotide is composed of sugar. What type of sugar? If it is DNA, deoxyribose sugar. If it is RNA, ribose sugar. So we have two types of sugars, deoxyribose and ribose sugar, which are pentose sugars, right? And second is what? Nitrogen bases. What are the types of nitrogen bases we have? Purines and pyrimidines. Purines and pyrimidines. Purines include adenine and thymine, guanine, sorry. Pyrimidines include cytosine, thymine, uracil. Right? After this, what do you have? Phosphate group. Right? So nucleic acid is a polynucleotide chain. It has multiple nucleotides. A single nucleotide is composed of sugar, nitrogen base, phosphate group. If it is DNA, what type of sugar? Deoxyribose sugar. What are the nitrogen bases DNA has? Adenine, guanine, thymine, cytosine. Uracil is not present in DNA. And then phosphate group. If it is RNA, what type of sugar you have? Ribose sugar. Nitrogen bases include adenine, guanine, cytosine, uracil. Good. And phosphate group. How they are connected you see. Okay. First you have to take the sugar, which is a pentose sugar. Here deoxyribose is given. C1, C2, C3, C4 and here C5 will be there. Okay. So, deoxyribose sugar, the structure is similar to ribose sugar, just at the second carbon OH will not be there, only hydrogen is there. Oxygen group is absent. That is the reason it is called as what? Deoxyribose. To the first carbon of this sugar, what is connected is nitrogen base. Where nitrogen base is attached to the first carbon of the sugar, right? And to the fifth carbon here, phosphate group is binded. Okay, with what bonds I will explain you in the next slide. So here you remember a single nucleotide C1, C2, C3, C4 and C5. To fifth carbon, phosphate group is binded. To first carbon, nitrogen base is binded. That is how a nucleotide is formed. Clear na till here? And these are the structures given in your NCERT. You have to fix them. Like how we have 3 amino acids. Okay, same like serine, glycine and methionine. We have 3, right? This also you have to fix it in your mind. Glucose, you see, it has 6 carbons. Ribose, it's given C1, 3, C4 and C5. 5 carbons. At second carbon, you can see oxygen group. So this is ribose sugar. If this oxygen is absent, then deoxyribose sugar. And here you see ma, nitrogen base. Purines are heterocyclic. It has 2 rings. You can drink water. Purines are heterocyclic. It has 2 rings. And how many nitrogens does it have? 9. Okay. So I said it is connected to first carbon. See, first carbon is connected to the nitrogen base adenine. So C1 of the sugar is binded to N9 of purine and C1 of the sugar is binded to N1 of pyrimidine. By what bond? N-glycosidic bond. Clear with the first bond ma? C1 of the sugar is binded to N9 of purine and N1 of pyrimidine by N-glycosidic bond. Next, C5 of the sugar is binded to phosphate bond by phosphoester bond. By what bond? Phosphoester bond. C5 of the sugar is binded to phosphate group, which is from phosphoric acid, H3PO4. Clear, right? So now how a single nucleotide is formed, you got an idea. What are the bonds involved? 2 bonds. What are they? N-glycosidic bond and phosphoester bond. Here you have to be noted with some terminology. Before that, we look into the amino acids once. See, adenine and guanine, heterocycles you can see. Here now the adenine and guanine, heterocyclic rings. Thymine, cytosine and uracil, homocyclic, single. They have some synonyms. This should be in everybody's book. Okay. Adenine is also known as what? 6-aminopurine. Adenine also addressed as 6-aminopurine. You can see here adenine. Sixth carbon you can see amino group. It is known as what? 6-aminopurine. Likewise, guanine is also known as what? 2-amino-6-oxypurine. 2-amino-6-oxypurine. 2 2-amino 6-oxypurine. Then cytosine also known as 4-amino 2-oxypyrimidine. Thymine also addressed as 5-methyl 2,4-dioxypyrimidine. Uracil 2,4-dioxypyrimidine. Right? Synonyms should be there with everybody. Please take down, and then we will go with the terminology as well. How a nucleotide is formed. What is the name you are addressing for a nucleoside? What is you are addressing for a nucleotide? Done? Once repeat, adenine is also known as guanine 2-amino 6-oxypurine. Cytosine 4-amino 2-oxypyrimidine. Thymine 5-methyl 2,4-dioxypyrimidine. Uracil 2,4-dioxypyrimidine. Clear no? Now you see only with respect to nucleotides for DNA and RNA. What is a nucleoside? So it is what sugar? Deoxyribose sugar. What carbon is attached to nitrogen base? First carbon of deoxyribose, no, be it ribose or deoxyribose, first carbon is attached to nitrogen base. What are the types of nitrogen bases you can see here? Adenine, guanine, cytosine, thymine. Okay. So these are known as deoxy. If adenine is attached, adenine nitrogen base is attached to the sugar, very good. Deoxyadenosine. Same the way, this is deoxyguanosine. And this will be deoxycytosine. And this will be thymidine. Somewhere I can hear thymosin. Okay, so all these are what ma? Nucleosides. Which carbon of this nucleoside is attached to phosphate group? Louder. Fifth carbon. To first carbon, nitrogen base is attached. To fifth carbon of the sugar, C5 is attached to phosphate group by what bond? Phosphoester bond. So when it is added with a phosphate group, this forms out what? Deoxyadenylic acid. Deoxyadenylic acid. Deoxy, tell me. Guanylic acid and deoxy correct, cytidyllic acid, deoxythymidylic acid. Here, any doubts? You can ask me. Which one? Anybody still in this slide? So here I have written this terminologies. 2 times in your NEET PYQs the question has come on this, which is very easy to answer, but then few didn't. Okay, so listen. Sugar is deoxyribose, nitrogen base is this, and the nucleoside you are addressing it as what? Deoxyadenosine, deoxyguanosine, cytosine, thymidine. Deoxyadenylic acid, guanylic acid, cytidyllic acid, thymidyllic acid. If you remember deoxy and keep ribose sugar here, this will be adenosine, guanosine, cytidine and uridine. In the place of thymine, what will be there? Uridine. Okay. And here adenylic acid, guanylic acid, cytidyllic acid, uridylic acid. Right? Please keep the terminology in your mind about nucleoside and nucleotide. So now we are clear on the formation of a single nucleotide. How a polynucleotide chain is formed? That is the second question. Can I move to the next slide? How a polynucleotide is formed. The linkage involved here. Listen carefully. So here, this is one nucleotide. In this single nucleotide, you are clear with this. C1 of this is attached to nitrogen base by what bond? N-glycosidic bond. C5 is attached to phosphate group by what bond? Phosphoester bond. Now this third carbon is free, right? OH group is free. And succeeding nucleotides, phosphate group is free. Okay? So this will be binded up with phosphodiester bond. How 2 nucleotides are connected by phosphodiester bond? If at all the question is how many bonds are involved in the formation of a single nucleotide? How many bonds are involved? 2. N-glycosidic bond, phosphoester bond. How many bonds are involved in the formation of 2 nucleotides or a nucleotide chain? 3. What are they? N-glycosidic bond, phosphoester bond, phosphodiester bond in a single nucleotide chain. For example, double-stranded DNA, how many bonds are involved? Hydrogen bond at the center. Like how? In proteins you have seen beta pleated sheets, right? Same likewise in DNA, when double-stranded DNA is formed, what connects the 2 nucleotide chains? Hydrogen bond at the center. How this hydrogen bond is connected to the nitrogen bases? Two nitrogen bases of 2 polynucleotide chains will be connected towards inner side by what? Hydrogen bond. Okay? If you remember, you remember. If not, write somewhere else. Single nucleotide, how many bonds are involved? Polynucleotide chain, how many bonds are involved? Double-stranded DNA, how many bonds are involved? If not, you will have time now only, you write along with me. This is polynucleotide chain. In shortcut I have written. Along with this 2, which bond is there? Hydrogen bond. Sorry, phosphodiester bond. And third, double stranded DNA. How many bonds are included? Along with this, hydrogen bond is involved. So if we have to see the number of bonds for a single nucleotide, how many number of bonds? 2. To form a polynucleotide chain, how many number of bonds are required? 3. Okay. To form a double stranded DNA, how many bonds are required? 4. Clear, no? Everybody? Done, ma? And then, so we'll also discuss the salient features of DNA here. Okay, if you are clear here, then it will be easy, the transition will be easy for NCERT 12. Let us recall. Okay, salient features of DNA. DNA, double stranded DNA has 2 polynucleotide chains which are running anti-parallel to each other. They are running anti-parallel to each other. One from 5' to 3', other from 5' to 3', but in opposite direction. Right? And they are complementary to each other. All the nucleotides present here are complementary to each other. And number of purines in the double stranded DNA are always equal to number of pyrimidines. Okay, so the first feature is what, ma? Anti-parallel or anti-polarity, or you can say anti-parallel to each other. Second is complementarity. Third, number of purines is equal to number of pyrimidines. Okay, that is adenine binded to thymine, guanine to cytosine. Number of purines equal to number of pyrimidines. Now comes the length of DNA. One turn is addressed as one pitch. Okay, one complete turn of DNA is known as what, ma? One pitch. So from here to here, is it complete turn of DNA, you tell me? Yes, right? From here to here, it is one complete turn of DNA. Okay, it is 3.4 nanometers or 34 angstroms. This is also known as one complete turn, one turn or pitch of DNA. And how many base pairs does a standard form of DNA will have? We have many forms of DNA, A, B, Z DNA. Standard form of DNA is B type DNA. It has around 10 base pairs in one complete turn. And the distance between two base pairs is 0.34 nanometers. Most of the students will get confused here. Okay, for one complete turn it is 3.4 angstroms. Sorry, 3.4 nanometers. But in between two base pairs it is 0.34 nanometers. Okay, this is for standard form of DNA I am telling you. Okay, and how many angstroms? 3.4 angstroms. Okay, these are what? Salient features of DNA. Anti-parallel nature, complementarity. Okay, and number of purines is equal to number of pyrimidines. One complete turn or pitch will have how much length of DNA? 3.4 nanometers or 34 angstroms, and distance between two base pairs is 0.34 nanometers. How you will calculate the length of DNA? Based on number of base pairs. Number of base pairs will give you the length of DNA. Got it? Now, so with this salient features of DNA is done. These are different forms of DNA you have to remember. Complete table not required. In this what you have to learn I will tell you. But listen carefully. First point ma, amongst these three types of DNA, which is more stable DNA which is present in the living organism is B form. That is the first point. Remembered? Which is most common in all cells. Remembered? And the helices, how they are winded up, in what fashion is important. B DNA is right-handed helices. A DNA is also right-handed. Z DNA is what? Left-handed helices. Third point is also clear, right? These both are right-handed helices and this is left-handed helices. Fourth point is number of base pairs. Number of base pairs in the B type DNA will be 10 base pairs. It is how many base pairs? 10 base pairs. A type DNA will have approximately 11 and Z type will have 12. Okay. In the rarest cases, we may expect question from major and minor groups. Major and minor group. Nothing but, see, there is more gap here, right? We'll take the example of B type DNA only. Can you see the gap here? This is addressed as major group. Can you see the small gaps here? This is addressed as what? Minor group. Like that. What is the major group here in B type DNA? Wide and deep. In B type DNA, major group is what? Wide and deep. In A type DNA, it is narrow and deep. Here it is absent. Okay. And here we go. B type DNA, minor group will be narrow and deep. This is wide and shallow, opposite. Easily you can remember, no? It is opposite. Next, this is same. Z type DNA is same that of what? B DNA. The 6 points you remember, it is enough. First is what? Stability. Which is more stable? B type DNA. Second is what? Most common in cells. Which is most common in cells? B type DNA. Third is what? Helices. What are right-handed helices? Which types of DNA are having right-handed helices? A DNA and B DNA. Which is having left-handed helices? Z DNA. Okay. And number of base pairs you see, B type DNA has how many? 10 base pairs. A will have 11 base pairs. Z DNA will have 12 base pairs. Major and minor groups you observe. Major group is wide and deep. Here it is wide and shallow. Minor group, sorry, here it is narrow and deep. Minor group wide and shallow. Here it is narrow deep. Same as that of B DNA. That's all. Okay. Clear to remember, right? Once please take down this in anywhere A, B and Z DNA. Just only that 5 points we have discussed, not all. Whether it is major or minor group. Done, right? So the Chargaff's rule, which we'll read in Mol Bio, which is connected with what? Salient features of DNA. Once we'll recall that here also. Erwin Chargaff has concluded that DNA molecule has how many? Amount of purine will be equal to amount of pyrimidines. Likewise, adenine is binded with thymine. I said you right. Nitrogen bases are connected internally by what bonds? Hydrogen bonds. Okay. Likewise, A double bond with T, and guanine is connected with triple bond to C. And there is something called as base ratio. Okay, you people said no, that is why I didn't repeat. There is something called as base ratio, that is A plus T by G plus C. This varies from species to species. For example, in prokaryotic cells you see, it will be less than 1. E. coli cells, for example, it has base ratio is 0.9. Eukaryotic cell, for example, human cell, the base ratio is around 1.3. Just remember that point enough. And then in deoxyribose sugar, phosphate components occurs in equal proportion. Like sugar and phosphate are equal. Number of sugars are there in the nucleotides, same amount of phosphate group will be there, right? Number of purines equal to number of pyrimidines. Clear with these 2 points, everybody? And there is one point to tell you. In the double helical structure of DNA, they will be winded up like this in the helical structure, right-handed fashion of the helix. Sugar and phosphate group will be towards the outer side. Sugar and phosphate group will be towards outer side, and nitrogen bases will be towards the inner side. Okay, with this we have exercised bonds in the nucleotide, polynucleotide chain, types of nitrogen bases, okay, double stranded DNA, salient features of DNA, types and forms of DNA, and then Chargaff's rule also. So clear na till here? And last slide of today's session, listen carefully, on something called as RNA. Okay, RNA we have 3 types ma. What are they? We have messenger RNA, ribosomal RNA, transfer RNA. Where are they synthesized? We have discussed also in cell the unit of life. If I assume this as a nucleus, inside a nucleus what we have? Nucleoplasm and nucleolus. Okay, inside a nucleolus what is synthesized? rRNA. By what polymerase? RNA polymerase 1. RNA polymerase 1. In nucleoplasm what are synthesized? tRNA and mRNA. mRNA by what polymerase ma? RNA polymerase 2. tRNA by RNA polymerase 3. Got it right? How much amount of these types of RNA are present? Messenger RNA is of 5%, ribosomal RNA 80% present inside a cell, tRNA around 10 to 15%, right? And the structure is same like that. How you will see the polynucleotide chain is same as that of what? Deoxyribose or deoxyribonucleotide chain, right? So with this we have completed 2 biomacromolecules. Those are what? Proteins and nucleic acid. In the next session, first we will start with objectives of this chapter till here, and then we will get on with what? Lipids and enzymes. Got it? So come prepared till here for the next class.